Hello and welcome to week 11. We've made it to gene expression, which is sort of one of my favorite weeks because we finally get to discuss the central dogma of biology. We get into the nitty-gritty, which explains how it all works. So we're going to start, and just for most of the section, we want to be thinking about what the vocabulary means in our regular lives and trying to make those connections with what the vocabulary means in class. So we're going to start with express yourself. If we were in class, I'd have you talk to the person next to you. What does express yourself mean to you? Hopefully, express yourself thinking about how you are outwardly presenting what you feel inside, what's your feelings and what makes you up, and how you present it to the world. And we're going to see that gene expression is the same thing. It's the process by which information in a gene, information in your chromosomes from your DNA becomes converted to RNA or ultimately protein. We're going to see that there's two things that are involved: transcription and translation. You are going to need to know the mechanisms of both of these. Now, in week 9, we went over DNA replication, where we saw how DNA went from being one double-stranded chromosome to being two sister chromatids. So DNA replication, DNA to DNA, which used DNA polymerase. That happened in the nucleus. Now we are going to be covering how we take that DNA message and convert it into RNA. We are going to see that that process is called transcription, DNA to RNA. The protein that does this is kind of similar to what we had previously learned, only instead of making many DNA, it's making many RNA. So it's RNA polymerase. So to make an analogy, we can think of it as the DNA is like an original blueprint for a building, where there's one file in one computer that's that blueprint. But then you make a copy of that and you print it out and you bring it to the construction site. That's this RNA. We're then going to see there's the next section, which is called translation. This is the process which converts that RNA into protein, into those amino acid polypeptide sequence that folds up to make a protein. We're going to see that this happens in and at a ribosome that is within the cytoplasm. The protein is the building. That RNA, that's what was used to build the actual building. We can think about it at that cartoon level I just showed you, or at the molecular level, where we have a DNA sequence with DNA nucleotides that through the process of transcription gets converted into RNA using RNA nucleotides. In the process of translation, we're going to see how 3 nucleotides gets converted into 1 amino acid. So to begin, I also want to think about the different forms of life which you might need to understand these processes in. We're going to see in bacterial cells, so in prokaryotes, DNA gets converted into RNA, gets converted into protein, all within the cytoplasm. Why is that? Because it's a bacteria cell and there are no organelles. There are no membrane-bound organelles. Everything happens in the cytoplasm in bacteria. Whereas in a eukaryotic cell, that DNA is protected by that nucleus, so therefore transcription, DNA becoming RNA, happens within the nucleus, whereas that mRNA message leaves the nucleus to find a ribosome or create a ribosome, and translation occurs in the cytoplasm. Now, the point of all of this is how we take the sequence of DNA, that's the heritable material, and how we make all this stuff, all those proteins. So we can do a quick review of proteins. To remember, protein's function is determined by its structure, and its structure is determined by the sequence of amino acids, because that sequence makes up the primary structure, which folds up based on the peptide bonds and the charges between the amino groups and the carboxyl groups, but then it all folds up depending on how big those side chains are, depending on what are their charges. Are they negative or positive? Do they have cysteines and will they form disulfide bridges? So we're trying to make proteins from genes. A gene is a specific sequence within the genome, within our chromosomes. So within one chromosome, there's many, many different genes. So on average, a gene is less than 10,000 base pairs. Some are smaller, some are longer. And we have an estimated approximately 20,000 genes total, which is actually a ton less than we had thought just 20 years ago. And it's important again to remind ourselves that most of the nucleotides within our chromosomes are not encoding a gene. So we got to remind ourselves again, there's some little differences between RNA and DNA. There's the difference in the sugar with that deoxyribose, and there's that difference in the base, the difference between thymine and uracil. And then, of course, either being single-stranded RNA or double-stranded DNA. If we were in class, we would have watched this video to begin with. So I'll just click down and show you something. I would encourage you to watch it if you want to appreciate the videos that will come after it. And what the individuals had to do to put this video together to try to explain transcription and translation. I'll also comment the guy at the beginning, Nobel laureate. I'd also encourage you to watch. This is a great overview, and it'll go through the process. So I am going to now spend the rest of this presentation going through what these videos are showing you. And it's important, though, to see it in this visual where you can watch the RNA polymerase, what we say by adding nucleotides to form those complementary base pairs. So another fantastic video to watch that's going to go through a lot of the same things I'm going through, but with different graphics. So the first step, transcription. What does transcription mean to you? Hopefully, it means something to thinking about what you're writing down. Transcript is a copy of all of your grades. So a transcript you make of someone talking, you're just writing down the words that you mean. In biology, it's the process where we copy DNA into messenger RNA. So it's this transcription, this first conversion. Like most things, or any good story, there's going to be a beginning, a middle, and an end of transcription. The beginning is called initiation. The middle is called elongation. And the end, termination. So beginning, middle, end. So what needs to happen for RNA transcription to begin? Well, we need to get the different proteins together on the gene. So we need to have the RNA polymerase start at the correct point. To find the correct point, there is a promoter. At that site, the helix is going to open up and RNA polymerase will be able to begin. So for that initiation complex to form, a few different things have to be found. One, there's going to be a specific part on that DNA. Remember, we have a huge amount of nucleotides. We need to find the right spot. So that's the promoter region. There's particular sequences in the DNA that define it as the promoter. That promoter region is going to have transcription factors, different proteins that are going to bind to the DNA in a different complex that are going to be able to recruit the RNA polymerase. So it's the transcription factors binding to the promoter that is going to signal the RNA polymerase, hey, start here. So RNA polymerase can bind. Then it will start adding RNA nucleotides at the start point. Just like we saw in DNA polymerase, RNA polymerase can only go in one direction, adding to those 3' end. There still is that 3' hydroxyl end that's ready to get a next nucleotide. So again, focusing in, we want to be thinking about how RNA polymerase is chugging along. Each base pair is being added according to those base pairing rules, where A's bind to T's on DNA, where A's in DNA bind to uracil, and C's and G's are always going to bind each other. There is direction to the transcript. RNA is synthesized from the 5' to 3' end. So that was initiation. Elongation is the middle. That's where the RNA polymerase is going to keep just chugging along, adding new nucleotides, and that RNA transcript is going to get longer. It's going to elongate. It's going to keep going until it reaches the end and termination. And what determines termination? The sequence in the DNA. And that RNA polymerase will fall off the DNA template. And you will have your completed mRNA transcript. Now, on one gene, you can have many RNA polymerases making multiple mRNA copies. So many polymerases can transcribe the same gene, and you can see that right here. Gene 1, gene 2, gene 3. So now we just talked about this picture, and we went over how RNA polymerase makes that copy from DNA to RNA. But if you remember this picture, there was actually a process in between, RNA processing, where that pre-mRNA gets all packaged up and cut up to be actually the RNA that can be used to make the protein. So we're going to look at RNA processing. So in words, or you can see here. So we have what's first transcribed, that's our primary mRNA. First, there's going to be a 5' cap of nucleotides added, and there is going to be a poly-A tail. What does that mean? It's the tail, meaning it's the 3' end, so it's the end, and poly-A, meaning it's a whole bunch of adenines, many A's. So the first two parts of RNA processing are that addition of a 5' cap and that poly-A tail. The last part is the removal of introns. As you can see right here, primary mRNA is much longer than mature RNA. Why is that? We actually cut out sections of nucleotide. The sections that are cut out are called introns. The sections that remain are called exons. And I'll fully state, it's a little bit weird. In many ways, I would want to say exons are excised, are cut out, they're exiting, introns stay in, but that is not true. Introns are sort of interrupting and they need to be removed. The exons are what stay. And this is important for many reasons. One, this cutting and pasting of our different genes together is a form of alternative RNA splicing. It can mean that we can have fewer genes and have some different products come out. And here you can focus a little bit more on how those introns are excised out involving the spliceosome. So we're not going to talk too much about the mechanism. Of course, there's different proteins involved, but it's the exons that remain in and it's the introns that are cut out. So that RNA processing involves that primary RNA getting a 5' cap, a poly-A tail, and becoming much shorter by removing the introns. Just have these little practices here. I'm going to assume you can pause your video, answer these questions, and then I got the answers on the next slide. We now talk about gene expression and doing that first stage of DNA to RNA, showing transcription. So the next part we have is translation. And in class, we'd have a brief discussion. What does translate mean to you? Hopefully, you're going to think of something to do with changing from one language to another language. So we saw transcription, that's just like copying down a transcript. Same language, just from written to oral. Translation, we see as switching from one language of nucleotides to the language of amino acids. So we've covered transcription, we've covered RNA processing, and now we're on to how does that sequence of nucleotides determine the sequence of amino acids. We can see that again, the RNA that was created is going to need to leave the nucleus to find a ribosome. So again, another fantastic video that's going to go through the same process I'm describing here, but with different visuals. So we have translation, this conversion of RNA to protein. This is going to occur in the ribosome, and it is going to involve multiple different types of RNA. So the three key RNAs for this level are rRNA, which is going to be making up the ribosomes, that's the R part. There's messenger RNA, mRNA, that's what you just heard about. That's that copy of a gene. And then we're going to learn about tRNA. tRNA stands for transfer RNA, and we're going to see that the tRNAs are going to be responsible for bringing in the correct amino acid to that ribosome. There's three different types of RNA that are going to be important in translation. So the first one is the one that we were introduced to. It's that RNA copy of the DNA. That's called messenger RNA. because it's the message from the DNA. We are going to see that that message gets read in a 3 nucleotide group called a codon. So a codon is simply 3 letters of nucleotides next to each other that are going to represent what amino acid is encoded for. So there's 64 different mRNA codons, and they each have 3 nucleotides. To use this chart, you find the first base, so in that case for here be U, here C, A, and G. The second base, that's the next one that you can read up here, and then you have the third base. So any different sequence can be found, and it's going to correlate to a particular amino acid. There's forms and tables that look like this. You can also use the wheel on the page in Canvas. The first nucleotide, your second nucleotide, and then the third nucleotide, and determine which amino acid is encoded for. And just quietly, this is remarkable. This is the code and the genetic code of life. The reason why you can have a bacteria express a protein from your human cells is because all of life uses the same genetic code. No matter if it's a bacteria or an insect, a horse or a tree uses the same genetic code. We are going to be using that genetic code to take that sequence of nucleotides, reading how 3 letters are going to get converted into 1 amino acid. This is going to happen at the ribosome. A ribosome is made up of proteins and rRNA. So I've got several more pictures of it that are a little less blurry, where you can see that there's 2 subunits. There's a large subunit and a small subunit. Prokaryotic cells have different subunits in terms of sizes than eukaryotic cells, but same thing, 2 subunits that are going to snap together. And they are going to have that mRNA sequence being the template for determining what amino acid is going to be brought in to the growing polypeptide chain. So we have the ribosomes made up of their 2 parts, made up of rRNA and proteins. So we also have tRNAs. Now T stands for transfer because the tRNAs are responsible for transferring the amino acid to the polypeptide chain. tRNAs are bringing the correct amino acids. How do they know they're the correct amino acids? Because a tRNA has 2 key parts. It has the part that's holding on to that amino acid, and it has this anticodon. The anticodon we're going to see, and the name suggests, is going to fit, is going to match, is going to have complementary base pairs to the codon in the mRNA. So there's an anticodon, and then there's the amino acid. You want to be able to picture it in different cartoons. There's these 2 key parts. So now we get to see how the 3 RNAs work together in translation, along with a few proteins. And just like we saw in translation, in transcription, we're going to see there's 3 phases to translation: initiation, elongation, and termination. So to start, what happens is that mRNA sequence comes out of the nucleus and gets bound by the small ribosomal subunit. It is going to bind when the initiator tRNA is going to recognize its anticodon to the start codon. The start codon is always the same, AUG. That's defining the start codon. The start codon's anticodon, UAC, is always going to be a part of this specific tRNA that will bring over methionine. Now that tRNA binding to that mRNA sequence is what recruits the large ribosomal subunit to form the complete ribosome. When the ribosome is completed and those 2 halves hinge together, we see that there's these different sites within the ribosome. There's the E, which is good because it stands for exit, you can think of. There's the P site, and there's the A site. So when that methionine binds and the ribosome comes together, that's going to be at the P site for the initiation or the start. And you can see the anticodon matches the codon. You can watch this video. Again, we have initiation forming that complex. I also want to point out this key part of this picture, which is also just reminding us this is going to involve energy. Just like we saw DNA replication, those nucleotides brought their own energy with them. For translation, we're going to need to supply that, and instead of using ATP, actually use GTP. Works just the same. It's that third phosphate bond producing the energy and producing GDP. So we have initiation, we're ready to keep going. We're going to see this is elongation. The videos kind of portray this a lot better where you can see the directionality of moving from 5' to 3'. But basically what happens in elongation is this new tRNA is going to always come in when it has the correct anticodon to match the codon within the A site, and it's going to bring in that next amino acid, and the P site is going to transfer that polypeptide chain to the amino acid on that A situated tRNA. This is going to require some energy, and it's going to require the peptide bond formation between those amino acids, and that chain is going to get transferred to the A site as mRNA is moving through, which ultimately then makes that A site become the P site as that ribosome continues right along. And this is this translocation step where that empty tRNA that no longer is holding on to that polypeptide chain will then be removed. And this whole circle keeps going as each new tRNA gets brought in and that polypeptide chain continues to lengthen. That ribosome is going to keep chugging along, bringing in the correct tRNA until it reaches a stop codon. So the stop codons are UAG, UAA, and UGA. So the ribosome will just keep going until instead of bringing the next tRNA that's holding another amino acid, it's bringing this release factor tRNA that produces the free polypeptide chain, and ultimately the ribosome will come apart, leaving that mRNA sequence and those two ribosomal subunits made up of rRNA and proteins. Of course, we can keep thinking about what actually happens to that polypeptide chain, and we can remind ourselves, but wait, weren't many of the ribosomes attached to the rough ER? Yes. How does that actually work? There's a signal peptide at the very beginning of that polypeptide sequence, which brings that ribosome to the rough ER, where that polypeptide chain is pushed into the rough ER. But of course, there's also free ribosomes where proteins are made just in the cytosol. And equally, just like we saw where one gene can have multiple RNA polymerase transcribing multiple mRNAs, we can see in translation, you can have multiple ribosomes on one mRNA sequence, making multiple proteins. So now overall, we've covered pretty much the whole process. You need to know transcription at the initiation, elongation, termination phase. You need to understand those three parts of RNA processing, adding the poly A tail and the 5' cap, cutting out all those introns, and following that mRNA sequence as it forms a complex with those rRNAs and tRNAs bringing in the appropriate amino acid to build that polypeptide chain. You need to know the different parts of the ribosome. And I'll also talk about here where you actually have a different enzyme where that amino acid gets attached to the correct tRNA. Again, it's an enzyme where the specific shape is going to make sure that the correct amino acid is bound to the correct tRNA that has the appropriate anticodon. So you can do a check yourself protein synthesis vocab fill in the blank. But here's actually even more important to be able to, can you use a sequence of the DNA template and figure out what would be that sequence of mRNA? What would be the anticodons in the tRNA? What will be the ultimate amino acid sequence? So you want to pause yourself and see if you can fill out this table. filling out where there's an A in mRNA, there's going to be a U in RNA, C's and G's, and you can fill out the rest of it. Now, to go from the mRNA sequence to the amino acid sequence, of course, you need to use the genetic code where you've got all the different amino acids represented. You have this special start codon and these 3 stop codons. So you can convert mRNA into the appropriate amino acid. There's a ton of practices in the lab and in the assignments. So another last check. Can you transcribe the information? Which enzymes carried out this process? So I've got all your answers here. You're again using your base pairing rules to talk about the different codons, which are again those 3-letter blocks. So the very last part of this presentation is to think about specific types of mutations. So in week 9, we talked about mutations where that DNA sequence gets altered. We saw before that structure, when something screws up the double helix, that can cause errors in DNA polymerase. We're going to talk about where there's just a single point mutation and DNA polymerase added just one different nucleotide, or there's some different types of mutations that can have broader impacts. So a nucleotide pair substitution is going to be when one nucleotide gets replaced for a different one. But there's actually different types of effects of this. You can have a silent mutation, where if we look here, if you change just that 3rd nucleotide, that's actually going to have no effect on the amino acid. They all encode the same amino acid, in this case proline. So that's a silent mutation, where it doesn't, you can't really see it, you can't hear it. There's a missense mutation, where you're changing one nucleotide again, but in this case, instead of it being CCC and making proline, maybe you switch to ACC and you have threonine. So it's still encoding an amino acid, it's just not the right one. But then you also have nonsense mutations. A nonsense mutation takes an amino acid and makes that codon into a stop codon, which will then, of course, lead to a non-functioning protein because the protein doesn't get to keep getting made. So these are examples of nucleotide pair substitution. We can have other mutations called a frame shift mutation, where instead of just swapping one nucleotide for another, you can actually insert a nucleotide and it's going to shift all the subsequent letters behind it. So this is called a frame shift, where it's not just one amino acid you're changing, you're going to change all subsequent amino acids. Examples of frame shift mutations are duplication mutations, where you can have, if you're reading those codons with the cat ate the dog representing 5 different codons, you insert a single E, well, you've screwed up all subsequent codons so that it's read differently. You can have a duplication, you can also have a deletion, which will also be a frame shift mutation. In this case, you're removing a nucleotide, or you're removing a larger section of the chromosome. You can also have inversions and translocations. So these are involving larger sections of the chromosome, but I'm still representing sort of in this way, where instead of having the cat, it gets flipped over, or a translocation where you're going to involve 2 chromosomes, but this is where you're swapping things. So these are different types of mutations that are all going to be able to have different degrees of an impact depending on what type of mutation they are. So again, we can do a little check where can you give yourself DNA sequence? These are all examples of point mutations. Can you make the RNA sequence and determine what's going to be the amino acid sequence? So which is going to be the most harmful, not harmful, or in between? Not harmful, a mutation that doesn't make any change? Well, that's not going to be harmful. We think about a mutation that's going to stop and make the protein fully non-functioning. Well, that's going to be the most harmful. And the result of this tryptophan, of this swapping, or of this missense mutation, well, that's going to depend. It's going to be somewhere in between. It might mean that it's fully non-functional, or it might actually be just as fine. So that is the end of the material for this week's section. I highly encourage you to go through the videos and watch the videos of how these proteins are moving, and then make sure you're quizzing yourself to be able to answer the appropriate questions. Good luck.